And Ahmed Dyer, and this is the TWIS Minion Science Island Hangout. Um, I'm hoping that Pamela Taylor can join us at some point. She's having problems with her internet connection through her smartphone. Until then, if and when she can get in, I will entertain you with a bunch of science stories. One is from NASA, and it has to do with the electromagnetic propulsion system they're trying to develop. And they're trying to create energy in a vacuum, because space is a vacuum. They did an experiment. They think they've been able to create some form of energy in this vacuum. They released the study for peer review, and there's a lot of skepticism about exactly what they produced. So I'll talk about that. Is an expert in the field answered some questions concerning it. Got another fun little story about the latest super material created by technology in the lab, and we're talking about silly putty. A bunch of researchers playing around with silly putty. There's some graphite over there. They don't know what to do with it. They throw it into the silly putty. The next thing you know, they're able to use it to monitor the pulse of one of the research assistants. That's a fun little story. Another fun little story about a bacteria that can produce a protein that in single cell eukaryotes, and we're a eukaryote, it can affect their sex drive. So who knows? Maybe we can use this little bacteria to produce an aphrodisiac that we can sell in China to those old Chinese guys all trying to get a hard on using rhino horn, and we can save the rhinos. Who knows? We've got another story about another piece of technology, and they're calling it mobile money, but it's using smartphones to, by women in Kenya, help them in their business, help to create start up their own new businesses, transition from crops over to some other sort of business, giving them some independence. Another story about another protein, a protein that produces an enzyme that can break into the biofilm that infectious agents, other bacteria, such as urinary tract infection, that creates a little biofilm to protect it from antibiotics. And now this enzyme can break through that biofilm. So that could be a good advance. Another laboratory that created an artificial kidney cells that function exactly like a kidney does. That is, it monitors, this is in mice, of course, it monitors blood sugar levels. It can deliver insulin when needed to these mice. Hopefully, somewhere down the road, it can have applications in humans. And the last story, if I get to it, concerns using cloud services for manufacturing companies using the cloud mainly for communications. So you've got a business and it's spread out all over the country, maybe all over the world. It was hard to get this communications going, and how the cloud computing is really facilitating these communications and helping companies to operate more efficiently. Okay, I'm gonna one more time to check to see if Pamela is able to get in, and not yet. Might have to send her an invite at some point. Okay, first story about NASA, and I am going to screen share it with you. You can get a look at this particular device they used to what they think is creating some kind of energy in a vacuum. And this is UConn Today. And they interviewed a professor, an expert in the field, Bruce Casanetti. And this is what he has to say about this particular propulsion drive and the experiment itself. I really don't know a thing about it. EM, an electric, EM stands for electromagnetic waves that produce thrust. The drive, which you can see a little picture of, consists of a truncated conical copper shield with a plastic disc covering the narrow end of the truncated cone. An electromagnetic wave is induced inside the copper shield in the same manner as a microwave oven. The propulsion system is unique because the device uses no traditional fuels or propellants. Instead, in the simplest terms, the electromagnetic waves bounce around inside the cone in a way that some say causes propulsion. In the NASA test, the thrusts produce something called 1.2 millinewtons. That's a really small amount of energy if it produced that. Very, very small but noticeable movement. The EM drive would make spacecrafts lighter and eliminate the need for massive amounts of fuel. And although the EM drive appeared to create thrust in these tests, there was no mass or any particles of any kind expelled during the process. And this is a violation of Newton's third law of motion, which is for every action, there's an equal and opposite reaction. Action and reaction is a direct result of the conservation of momentum. The violation of such a basic law is as the conservation of momentum would invalidate much of the basis for all of the physics as we know it, Newtonian physics. Hence the skepticism by a lot of scientists and engineers. They think that, maybe even some sort of experimental error. Adding to this is the fact that those who believe the results are valid do not yet have an experimental way of validating or proving this physical explanation and a theoretically plausibly proven physical explanation. This particular expert, this Bruce Casanetti, believes that there is some mundane explanation for the results. For example, electric currents are heating components within the drive that expand during the experiments, causing motion that would appear as a force. If the results are valid, it points to new physics. Newton's laws have already been shown not to apply at high relative speeds where special relativity applies, or in large gravitational fields and with small cycle molecules. But Newton is mostly right. There are certainly many aspects of physics we do not understand, and some aspects so mysterious we don't even know where to begin. If EM drive is tested in space, then the acceleration could be directly measured, which would eliminate all of the confusion associated with force measurements. Space would provide an ideal vacuum, as the device would not have to be placed in a vacuum chamber and provide a weightless environment, eliminating any need for support. But space missions are very expensive, and it costs $10,000 to launch 1 pound of material into orbit. It might be better to try to do it experimentally if in fact they can do it. So what you have is an interesting experiment. Something happened in this vacuum but we don't know exactly what. Um, so who knows? Hopefully, I'm hoping, we're hoping that NASA is able to use their propulsion drive, their electromagnetic propulsion drive in space. It would be a huge advance, but a lot of skepticism about the results of this particular study, and even can they replicate it. Okay, going on to another fun study, we're talking about silly putty for science. And I'm going to try to screen share for you again. Hopefully it's working for you. There you go, silly putty. Okay. Yeah, the dash of graphene can transform silly putty into a pressure sensor able to monitor a human pulse or even track the dainty steps of a teeny tiny little spider. The material called G-Putty could be developed into a device that continuously monitors blood pressure. It also demonstrates a form of self-repair that may herald smarter graphene composites. Since graphene was first isolated in 2004, researchers have added these atom-thin sheets of carbon to a whole bunch of different materials, hoping to create composites that benefit from its superlative strength and electric conductivity. But there have been surprisingly few attempts to blend it with a viscoelastic material such as silly putty, which behaves as both an elastic solid and a liquid. Leave a lump on the top of a hole, for example, and it will slowly ooze through. Connor Boland, a researcher working at the Jonathan Coleman's Nanotechnology Lab at Trinity College in Dublin, wondered what would happen if he brought the two materials together. They mixed graphene flakes, roughly 20 atom layers thick and up to 800 nanometers long, with homemade silly putty to produce a dark gray G-putty that conducted electricity. Crucially, its electric resistance changed dramatically when the researchers applied it even to tiny amounts of pressure. The putty was at least 10 times more sensitive than any other nano composite sensor. When they wired it up to a student's neck, the pulse from his carotid artery was clearly visible in these resistance changes. In fact, the pulse profile was so detailed that they could convert it into an accurate blood pressure reading. The sensor also monitored respiration when placed on the student's chest. And as a slightly bizarre encore, it recorded the individual steps of a spider weighing less than 20 milligrams. Coleman's team found that a graphene flakes form a conducting network within the putty, and deforming the material breaks the network apart, rapidly increasing its electric resistance. G-putty's low viscosity then allows the graphene flakes to move back into position and reform the network. It's a self-healing phenomenon. Coleman is already talking to medical device companies who are interested in using G-putty for continuous physiological monitoring. Blood pressure measurements, for example, often rely on bulky cuffs around a patient's arm and often only a snapshot reading. A cheap, non-invasive sensor could provide a simple way to monitor patients at home. G-putty would have to clear a lot of hurdles. obviously, including proving it can be made in large-scale quantities and in real-world testing to assess its long-range performance before it could be commercialized. For real applications, you need to have it work the same way thousands of times. All right. Come on, screen share. There you go. So, that's amazing. A little silly putty. There you go. Little lab experiment. Bunch of people throwing some graphene into some silly putty, and bang, you got yourself an extra sensitive sensor. And just use something you could just kind of wrap around your arm, paste a little, some little G putty on your chest or something, and there you go. 24 health monitoring. That would be fantastic. I'm gonna let me see. Let me check. Back on get back on Facebook. Okay, haven't heard from Pamela yet. All right. And another fun little study talking about a bacteria that can affect the sex drive of a eukaryote. All right. Let me do the old screen share again. All right, here you go. You got a frenzy. That little picture, if you're seeing it, and hopefully you are, you got a frenzy going on, a sex frenzy. Okay. Reading from the study. No, reading. That's not the right one. Okay, Ed, get the right one. All right. Scientists have demonstrated that bacteria can drive mating in eukaryotes, raising the possibility that environmental bacteria or bacterial symbionts may influence mating in animals. And this is from the American Society of Cell Biology. Researchers seeking the evolutionary roots of the animal kingdom have discovered a bacteria. It's called a Bioflim that acts as an aphrodisiac when releasing an enzyme that sends the, coenflagellate, something called Sapphilin rosetta, one of the closest living relatives of animals, into a mating frenzy. This co-flagellate, or something like that. I got no clue how to pronounce that word. It's a eukaryote, just like we are. Their cells have a membrane-bound nucleus containing their genetic material, and they live free as single cells in multicellular colony. Researchers Nicole King and Aurelie von Wozniak of the University of California, Berkeley, discovered that in minutes after exposure to a chondrite sulfate, CS, produced by this V. fisheria, Rosetta cells aggregated into mass mating swarms, entering into cells on nuclear fusion with duplicating and recombining their genetic material. One of the first demonstrations that a bacteria can drive mating in eukaryotes. They add that this discovery raises the possibility that environmental bacteria or bacterial symbionts may influence mating in animals as well. In recent years, the study of human biology has been shaken up by discoveries how bacteria that live in the gut, the so-called microbiome, affect metabolism, the immune system, and disease progression. The tangled symbiotic and pathogenic relationships between bacteria and multicellular animals go back into the deep evolutionary time, where fossils of ancestral microscopic soft body eukaryotes are unlikely to have survived. But living in these conoflagellates, King believes researchers have a model organism to compare with animals and possibly dissect the roots of animal multicellularity. All right, there you go. Ending the screen share. The first thing that occurs to me, of course, I think of the old guys in China, mostly in China, probably some in the United States, and they're killing rhinos just for their horn because they believe it's an aphrodisiac. Now, man, if we can get this bacteria, and if we can take the enzyme that they're able to produce, and if it can work with the human animal, bang, there you go. You sell it on the market, and maybe we can get people off using rhino horn and help save the rhinos. All right. Got another story about another medical advance, and this is periodontal disease. Now, because of my Celtic background, I believe, I have a predisposition to periodontal disease. I have these deep pockets in between my teeth, makes it harder to clean, easier for bacteria to hang out. And in my family, I'm the only one over the age of 60 who doesn't have a full dental plate. I got a partial plate for a couple of teeth that I've lost, but so far, I've been able to save all the rest of my teeth. You know, I floss all the time and I brush and all the rest of it. Sooner or later, I'm probably going to need a full plate because, as I say, this dental disease, I just have a predisposition towards it. Now, let me go to the study. Let me screen share. Okay, hopefully that worked. And reading from the study, a world-first vaccine that could eliminate or at least reduce the need for surgery and antibiotics for severe gum disease has been validated by new research. And this is from the University of Melbourne in Australia. A world-first vaccine developed by Melbourne scientists has been validated by research published this week. A team of dental scientists at the Oral Health University of Melbourne has been working on a vaccine for chronic periodontitis for the past 15 years. Clinical trials on periodontal patients could potentially begin in 2018. The analysis of the vaccine's effectiveness by collaborating groups based in Melbourne and at Cambridge in the US. The vaccine targets enzymes produced by the bacteria Prophoromonas or something like that, gingivitis, to trigger an immune response. This response produces antibodies that neutralize the pathogen's destructive toxins. Gingivitis is known as a keystone pathogen, which means it has the potential to distort the balance of microorganisms in dental plaque. Eric Reynolds said it was hoped the vaccine would substantially reduce tissue destruction in patients harboring gingivitis. We currently treat periodontal disease with professional cleaning, sometimes involving surgery and antibiotic regimens. Now, in my case, I did go through antibiotics. Works temporarily, but in the end, it came back. And I've gone through the dental cleaning and everything else. There's a procedure that basically means cutting out all of the diseased tissue, which would be all of the tissue around my upper and lower gums, and trying to replace that with tissue, I think, from around the cheek, and then hopefully it will grow. Now that whole process is going to cost $4,000. I ain't got $4,000. So I'm hoping that the teeth cleaning every 6 months, my flossing 3 times a day will do the trick and put off having to replace any more teeth. Okay. These methods are hopeful, but in many cases, the bacteria reestablishes in the dental plaque, causing a microbiological imbalance, so the disease continues. There isn't a lot of detail in this first story, so I went to the abstract. The abstract is 35 pages of PhD talk. I got no clue what they were talking about. Now, I do have part of the abstract. I'm going to read it to you. Maybe it will help some of you out there understand what we're talking about. Now, from the abstract, it says this gingivitis infected mice with an established inflammatory immune response were protected from developing bone resorption by therapeutical vaccination with what they're calling a chimera, KSI2-A1, an immunogen targeting the major virulence factors of the bacterium. Protection was characterized by a antigen-specific antibody and the T2 cell response. Adoptive transfer of this KAS2A specific Ig antibody, I'll call it, expressing B cells, confirmed that Ig1 mediated protection. Furthermore, prenatal or interoral administration of KAS2A1 specific antibodies protected against the development of gingivitis, bone resorption, loss of bone tissue. Specific antibodies neutralize the gingivitis by inhibiting proteolytic activity, binding to host cells, etc. You get the idea. Hopefully, once again, that word hopefully, and this is in mice, this is a mice study. It can be developed to help with humans, but something like, we're still mice study, we may be a decade away before it would actually have applications for humans. You got to go through clinical trials. So a hopeful first step. Let me check. See you. Yeah, I guess Pamela's not going to make it tonight. Okay. Going to talk about another medical advance. In this particular case, I'm talking about a protein that it's an enzyme that can break through the biofilm of a bacterial infection, and the biofilm protecting the bacteria from antibiotics. All right, let me do the screen share thing. again. There's a pretty little crystalline structure of this protein complex. Onto the story. Researchers have discovered a protein that inhibits biofilms of a bacterium responsible for many cystic fibrosis infections. This is California Institute of Technology. Many infectious pathogens are difficult to treat because they develop into biofilms, layers of metabolically active but slowly growing bacteria embedded in a protective layer of slime, which are inherently more resistant to antibiotics. The researchers at University of Oxford and Caltech have made progress in the fight against biofilms. Diana Newman, she's a professor of biology and geobiology at Caltech, identified a protein that degrades and inhibits biofilms of something called Pseudomonas aeruginosus or something like that. It's the primary pathogen in cystic fibrosis infections. And this, I'm going to call it PA, causes chronic infections that are difficult to treat, such as those that inhabit burn wounds, diabetic ulcers, and the lungs of individuals living with cystic fibrosis. In part, the reasons these infections are hard to treat is because this PA bacteria enters a biofilm mode of growth, and in this context, the biofilms tolerate conventional antibiotics much better than other modes of bacterial growth. The group targeted, and this was a piocinin, a small molecule produced by PA bacteria that produces a blue pigment. The molecule has been used in clinical identification of this strain for over a century, but several years ago, the Newman group demonstrated that the molecule also supports biofilm growth, raising the possibility that its degradation might offer a new route to inhibit biofilm development. To identify a factor that would selectively degrade piocillin, a researcher by the name of Kyle Costa turned a milligram of soil collected in the courtyard of the Bachman Institute of Caltech campus. This is from soil. From the soil, he isolated another bacterium that produces a previously uncharacterized small protein called piocillin. Adding this, he calls it pod A, to growing cultures of the bacteria that produces periodontal disease, the team discovered it inhibits biofilm development. Well, there was a precedent for the use of enzymes to treat bacterial infections. The novelty of this study lies in our observation that selectively degrading a small pigment that supports the biofilm lifestyle can inhibit biofilm expansion. What is interesting about this results from an ecological perspective is that a potential new theoretical approach comes from leveraging reactions catalyzed by soil bacteria. These organisms likely co-evolved with the pathogen, and we may simply be harnessing strategies other than microbes to use to keep a check on nature. That's pretty cool. A bacteria in soil that we may be able to use to in place of antibiotics. That'd be terrific, because antibiotics have all kinds of side effects, and usually what happens is one round, if it's not completely effective, that second round isn't going to help you because all you've done is created super germs. So now if they can come up with an enzyme that can break down the biofilm protecting the bacteria infection, fantastic. I got a real surprising study that surprised me. In 2015, the life expectancy in the United States actually went down. A very small amount, but it went down for the first time in over 20 years. Okay, come on. Let me drag it down here. Okay. Now, reading from the study. Reading from this article in the Christian Science Monitor. Life expectancy in the United States. Now, let's take, here's a picture of some people bicycling. Life expectancy in the United States dropped in 2015 for the first time since 1993, according to the Center for Disease Control and Prevention. The average life expectancy at birth decreased by 0.1% of a year compared to 2014. That's about a month. While the change is not particularly large, the drop came as a surprise to many researchers, considering the generally upward trend in life expectancy over the last few decades. I mean, that's what I expected to see. I mean, you get these tremendous advances in medical research, in healthcare, that really we should see a continuous line upward. And now we got a little blip in that trend. The recent drop reflects a number of different causes. Actually, well, the researchers think they know the primary reason why this drop occurred, and they call it diseases of despair. The opioid overdose epidemic, suicides, reflecting a lack of economic opportunity and hopelessness among white working class males. And it's just a guess. I don't know how you go about proving it, but I mean, we're getting all these advances, so why isn't our life expectancy continuing to increase? And, you know, I've read about the opioid epidemic that's happening in the United States. I did not realize it'd be that extensive to cause a lower birth rate overall. Let's see. This is unusual. This is the guy. His name is Jacquin Zeus, XU. He's an epidemiologist at the Center for Disease Control. 2015 is kind of different from every year. It looks like much more depth than we have seen in a few years. Much more death, excuse me, than in a few years. Of the 10 leading causes of death, 8 increased in 2015. 1 stayed statistically the same, and 1 actually decreased. Infant mortality rates, which are generally regarded as a good indicator of the overall health of a population. According to the report, was basically unchanged from 2014. Since there's no smoking gun and the drop in life expectancy last year's, experts cite a number of issues that should be addressed. So we've got almost an across-the-board increase in deaths. But beyond treatment of diseases, there is need to address income inequality and unemployment as well. And what they're saying is that's the primary drive. Income inequality and unemployment is causing more to turn to opiates, drugs, increasing depression, leading to increased suicides, poor nutrition, and drug overdoses. Let's see, the paper was presented to the Proceedings of the National Academy, found that the surprising rise in middle-aged white mortality in America from 1999 to 2013 was driven by a rise in suicide, drug abuse, and alcoholism. The trends were strongest among those with the least education and in the predominantly red South and West, where the authors suggesting a vicious cycle of physical pain and addiction to painkillers compounded by physical uncertainty. They don't know if they're going to, you lose the job, you don't know if you're going to be able to get another job. Now, it's a bit of a leap to pinpoint, to put a political blame, talking about red states versus blue states. I don't know. I think it's a bit of a leap. It could be a connection. Now, a guy by the name of Mark Rank, he's a sociologist at Washington University in St. Louis. Now, what he's saying is the Republican push to cut government spending and help businesses have meant more risk has been shifted from employers and government to the backs of individuals and families. This means we need to shift our framework from thinking that it is them to thinking it is us, that it is, in fact, it's middle America that is really experiencing some hard times. The struggle of young people without a college degree in rural towns to find full-time work is a symptom both of economic and cultural ties. Work habits dull for the unemployed. Young, idle men struggling to find life partners or even to get their own place in society. They're turning to drugs and alcohol more frequently. The CDC is saying we're in the midst of an opioid overdose epidemic. Wow. That's, it's really hard for me to believe that this epidemic could be so great that it's actually causing a drop in life expectancy for the first time in 22 years. I mean, that's mind-boggling. I mean, at one time, wasn't that many years ago, Americans were the most optimistic people on the planet. And now, looking at studies like this, what we're seeing is, working class people, ordinary working class people, and now they're having a hard time finding work. And if they're finding work, it's maybe just less than minimum wage job, not even making enough money to, to put a roof over your head and food on the table. I guess it is really, it's an epidemic, drug overdose epidemic. Wow. So we've got this symptom, a drop for the first time in 23 years in life expectancy. That's a wake up. That should be a wake up call that something needs to be done. And I don't think local communities certainly will try to address the problem. I do read about that. Local programs trying to reach people who are depressed, trying to reach the addicts, trying to get them to turn around lives. A lot of local programs, obviously not enough. not enough at the local level, not enough at the state level. The only agency with the resources to address the problems, federal government. Unfortunately, we're likely to be in a period of at least 2, maybe 4 years where the federal government is going to be providing less support, not more, but less support for people. And I guess the idea is, you lower business tax, business will employ more people. Maybe that will happen. Hope it happens. And by, providing more employment for people, this will address this issue. I don't know. And it's income inequity again, which means even if you get more jobs created, they don't really pay enough in so many cases to put a roof over your head, food on the table. And you think about the future. What kind of a future have you got? What, your kids are going to be flipping hamburgers all their lives? I don't know. It's a symptom. We got to do something about it. And it doesn't look like we will. And I'm such an optimist usually. Oh, well. Okay. Now we do have some people who are helped by technology. And these are women in Kenya. And let me do the screen share thing once again. And let's see. Okay, there you go. Happy smiling face of a woman in Kenya. And she's thrilled because she's now selling her products using this mobile money. That's not really the word. It's what you've got is in Kenya what amounts to a digital bank, and it's a means for people to sell their products online, people to pay for those products online. And it's really helping and facilitate businesses for these women. Now, reading from the study, since 2008, an MIT economist, Tavnit Suri, he's been studying the financial and social impacts of Kenya mobile money services, which allows users to store and exchange monetary values via mobile phone. And her work has shown these services have helped Kenyans save more money and weather financial storms, among other benefits. So she produced this Tavnit Surya. She produced a new paper showing that mobile money services have had a notable long-term effects on poverty reduction in Kenya, and especially with women. The study estimates that since 2008, access to mobile money services increased per capita consumptions by a level of 2% of Kenyan households. So it raised the level of consumption in 2% of Kenyan households, lifting them out of extreme poverty, many of them making less than $1.25 a day. And interestingly, female-headed household saw far greater increases in consumption than male-headed households. And they're estimating that 185,000 women moved from farming to business occupations. 185,000 women. Wow. By 2015, more than 270 mobile money services were operating in 93 countries, an estimated of 411 million accounts. The Kenyan study is important because it shows that mobile money services aren't just conveniences, but do in fact have a positive impact on people's livelihood. The study works its Empower M-PESA, M-P-E-S-A, the country's most popular service, which was launched in 2007 and now services 25 million Kenyans. There are more than 120,000 M-PESA agents scattered throughout the country. They showed that this M-PESA, P-E-S-S-A, helped users borrow, save, and pay for services more easily. 2012 study showed M-PESA helped Kenyans manage financial uncertainties caused by crop failures, droughts, and other health issues. Users can use a wider network of support and receive payments more quickly. See, going on, instead of looking at the number of individuals, they studied how many of these agents there were, how many of the agents there are signing up customers. And I'm going to skip a whole bunch of stuff. And the researchers estimated that the spread of mobile money services has helped 194,000 Kenyans overall and induced 185,000 women to work in businesses or retail occupations over farming. They can become more independent. Let's see, the researchers also think mobile money can give women in male-headed households or usually secondary income earners more financial independence. Women sometimes they are not able to save on their own because cash gets used by the whole household, controlled by men in most cases. It allows them to keep their cash separate. Mobile money. Now, I personally have never used it. I never used my smartphone to buy anything. I have certainly gone online, and the majority of my purchases are online, except really for groceries. Groceries, I'm going to the supermarket, and there I'm using a kind of mobile paying device, but I'm using my credit card. So I'm actually using my credit card 90%, 99% of the time to buy something. But even that operates through the cloud, so to speak, operates through the internet. I'm accessing my banking accounts through the internet. That's kind of a cloud service. Obviously, it's going to increase. And these third world applications, you have women, and they want to become independent. They want to maybe save some money on their own. Well, now, using these digital banks, they can more efficiently, more easily reach their customers. Everything is done, through the internet, and they can save some money. So, you know what I'm talking about. So it's. I don't really know. In the long run, it's obviously going to have a tremendous effect. It will continue. It's having a tremendous effect now. It will have tremendous effect in the future. It's decreasing brick and mortar stores. Will that help? Will it help increase unemployment? Will it help people find more jobs? I don't know. Hopefully, it will help. It will produce more jobs than it's ending. Who knows? All right. What else we got? What else I got on the agenda? Got an interesting little study, and we're talking about creating artificial kidney cells. Now, I'm a diabetic, so I don't need insulin at this point. I can control my diabetes through diet primarily. But I have a lot of friends who need insulin, and if they can create artificial kidney cells, and perhaps at some point in time down the road use them, I don't know, implant them in their body or whatever, take these kidney cells that are not working properly, instead of using stem cells, but use these artificial kidney cells, which according to the research think will be a big improvement. That really would be a great help. Okay, let me do the screen sharing thing again. Here we go. There we go. There is a picture you are hopefully looking at of this little designer cell, artificial kidney cell. Now this is from the ETH Zurich. This is Martin Fusenegger of the Department of Biosystems Science and Engineering, and they have produced artificial beta cells using just a very straightforward engineering approach. The artificial beta cells can do everything that natural ones do. They measure the glucose concentration in blood and produce enough insulin to effectively lower the blood sugar level. For the new approach, the researchers used a cell line based on human kidney cells, HEK cells. The researchers used natural glucose transport proteins and potassium channels in the membrane of the HEK cells. They enhanced these with a voltage-dependent chemical channel and a gene for the production of insulin and GLP-1, a hormone involved in the regulation of blood sugar. The artificial beta cells, the HEK cells, natural glucose transport protein carries glucose from the bloodstream into the cell's interior. When the blood cell's sugar level exceeds a certain threshold, the potassium channel closes. This flips the voltage distribution at the membrane, causing the calcium channels to open as calcium channels flow in. This is why I need somebody in the Hangout to help me. As calcium channels flows in, it triggers the HEK cells built-in signal cascade, leading to the production and secretion of insulin. The initial test of the artificial beta cells in diabetic mice revealed the cells to be extremely effective. They worked better and for longer than any solution achieved anywhere in the world so far. When implemented into diabetic mice, the modified HEK cells worked reliably for 3 weeks, producing, the old man's brain cells are not working. The longer we go, the harder the brain-tongue interface is working. Okay, providing sufficient quantities of the messengers to regulate blood sugar level. In developing the artificial cells, the researchers had the help of a computer model created by another group headed by Jorg Stilling. The model allows predictions to be made of cell behavior, which can be verified experimentally. Okay. He and his group have been working on this biologically based solution for a long time. Several months ago, they unveiled beta cells that had been grown from stem cells from person's fatty tissue. But that technique is expensive, and that's the key here. This would be a cheap, inexpensive way of creating artificial kidney cells. And I'm going to stop there because you get what I'm saying. All right. Now, unfortunately, rats. I'm really hoping that somebody else would get in here to help me out here. Let me see. No, Pamela. No, Justin. Okay. Well, I kind of reached the point where I'm getting tongue-tied. I really hope I provided you with some interesting information. I'm going to have links in the show notes, so you're going to be able to follow up and read the stories if you want to. And hopefully next week, I think Pamela's been using her smartphone to try to get into the Hangouts. Didn't work this week. Hopefully next week she'll be moving to an area where she'll be able to use a better Wi-Fi connection, be able to use her computer. I don't know, maybe she's got a laptop. In any event, I'm going to sign off because I'm getting a little bit kind of tongue-tied here. There's nobody in the Hangout. I can get a real discussion going on. So I just don't want to sit here reading stories to you, and I pretty much made it through the entire lineup. So hopefully next week, some other people will join me. Yay! And we'll have a more interesting Hangout. So in any event, we got a viewer. Sorry, viewer. Hopefully you'll be able to go on to the YouTubes and look at the first part of the Hangout where I can talk reasonably well, and you'll be entertained and you'll be informed. So good night. I'm stopping the broadcast.